Welcome back. Today we are going to look at the lymphoid cells. Lymphoid cells come from lymphoid stem cells and they are referred to as lymphocytes. We saw this is because they reside mainly in the lymphatic system. There are three types: T cells, B cells, and null cells. The general structure, they look like this, we explained earlier. They are spherical or nearly spherical with a large nucleus and narrow cytoplasm. They also lack visible granules under light microscope, except the null cells, which have granules. So general structure, I have said they have large nucleus, narrow cytoplasm. On their surface, they are receptors. They have main receptors depending on the type of lymphocyte. Then there are those we call co-receptors also on their surfaces. This one are classified as CD receptors, so which stands for cluster of differentiation. You'll see why they are called cluster of differentiation. So on their surface, we have main receptors for each of those type of cells. Then they are also co-receptors. General characteristics: lymphocytes are mononuclear cells. That means they have single cells. Remember, neutrophils are the single cell which is lobed. These ones are the nucleus is not lobed. So they are mainly involved in adaptive immunity, except null cells, which could also be involved in immunity. So they are second most abundant leukocytes. So they are found in blood cells and lymphoid organs mainly. They are motile. I say they derive the name because they are closely associated with the lymphatic system. So they are long-lived as compared to neutrophils. They undergo differentiation, maturation, and education, that's sensitization, when they are activated by antigens. They form from bone marrows as lymphoid stem cells. In fact, they have large nucleus and narrow cytoplasm. T and B cells are specific, null cells are not specific. So let's look at subtypes of lymphocytes. I've mentioned up there are three: B cells, T cells, and null cells. So let's start with B cells. So B cells produce antibodies. They secrete antibodies, that's their function. And on their surface, the main receptors are antibody molecules. The main receptors are antibody molecules. So they make up some co-receptor CD, but we are not going to go into that today. So when they are stimulated by antigens, they differentiate to form plasma cells. Plasma cells is a form of B cells which secrete antibodies. B cells must change to plasma cells after antigen stimulation for antibody to produce. So it is plasma cells produce antibody. The other group of cells form B memory cells. These are cells which have been exposed to the antigen and memorize the antigen. So in the second infection or attack by the antigen, they react very fast, at a very fast speed. So what is the role of B cells? They produce antibodies against specific antigens. They act as antigen presenting cells just like macrophages. They also process antigen and present them to T cells during activation. Then let's look at T lymphocyte. T lymphocytes have T cell receptors on their surface. Normally we refer to it as TCR, T cell receptors on their surfaces. And they are the ones which are majorly known for having quite a good number of co-receptors, the class of differentiation. So this class of differentiation has been used to classify them into different subsets. So you can see here the CD4. So class of differentiation 4. So the main class of differentiation are CD4 and CD8. So most T cells could be known to have CD4 on their surfaces, a co-receptor, and CD8. So this has been used to classify the T cells, although loosely. So the T cells we have, they are those which regulate immune system and the ones which normally kill cells, infected cells or abnormal cells in the body, or suppress the immune system. So this group have CD8 plus TCR, that's T cell receptor on their surfaces. This group have mainly CD4 as co-receptor and TCR on their surfaces. So let's look at the CD4 cells. The first group of the subgroup of or subset of T cells is called T helper cells, abbreviated TH, or just CD4 cells. Sometimes called regulatory T cells or Tregs, reg for regulatory. So the regulatory T cells sometimes are grouped different from T helper cells, but normally their functions is more or less the same, regulating the process of adaptive immunity. So T helper cells normally help in antigenic stimulation of B cells to produce antibodies and other functions. So they also group two groups of T helper cells. There's T helper 1 and T helper 2. So T helper 1 release cytokines, in this case lymphokines, for the T helper cells are lymphocytes. These lymphokines that mediate or regulate immune response. But T helper 2 stimulates B cells, develop plasma cells to produce antibody. Then we have T memory cells, which once exposed to antigens remain in the body for quite a long time to memorize the antigen. So the next time the antigen enters the body, they react very fast without a vigor to protect the body. Then we have T inducer cells. As the name suggests, this one trigger maturation of T cells from stem cells. Then we have the CD8 cells. The first one is cytotoxic T lymphocyte or T cytotoxic cells, normally just called CD8 plus cells. This one kills target cells, virus infected cells or tumor cells by cytotoxicity. Then we have suppressor T cells, which inhibit antibody production. Then we have T delayed hypersensitivity cell, which take part in delayed hypersensitivity that we will discuss much later. So as we have said, and we will continue repeating, T cells are mainly of two types. We have CD4, which regulate the immune system and coordinate it, and CD8, which mainly do cytotoxic killing. So main functions of T cell, they trigger or induce B cells to mature to plasma cell and produce antibody. They recruit and activate other cells of immune system, that's mononuclear cells and phagocytes. They secrete lymphokine responsible for growth and differentiation of a set of immune cells, and they regulate immunity. So then we look at null cells. Null cells are called null because they are not specific and they lack the common characteristics of surface markers B and T cells. So they have markers but not the same as the one found B and T cells. They are lack both specificity and memory. So they are not specific and they don't have memory as B or T cells. So we have two types of null cells. We have natural killer cells. Natural killer cells. So this is the group of null cells which their function is to kill by cytotoxicity. They kill target cells by cytotoxicity. They have large granules in the body. No, this one I didn't mention, but I mentioned that null cells, apart from non-specific, they have granules. So they are only granulated lymphocytes, and they are larger than B or T cells. So they are referred to as large granulated lymphocytes. So they also, I'll say there are two types. So we have the natural killer cells, which are CD16 as the co-receptors. The nucleus, they are kidney shaped and they kill target cells by cytotoxicity. The cytotoxicity is not antibody dependent, it is antibody independent. So they destroy cancer cells and virus infected cells. So then we have killer cell. Now this is not natural killer cells. This one's also carrier cell cytotoxicity, but antibody dependent cell cytotoxicity. They also kill tumor cells and cells which are infected by intracellular virus, bacteria, and the rest. So this one carry out cell cytotoxicity but antibody dependent. This one carry out cell cytotoxicity but without depending on antibody. Thank you, and let's meet another time.